So we're very pleased to have Professor Michal Ben-Shachar with us today. Professor Ben-Shachar is a professor in the English department and a member of the Gonda Multidisciplinary Center in Bar-Ilan University, Multidisciplinary Brain Center in Bar-Ilan University. She's the head of the Neurolinguistics Lab. She completed her MA in cognitive psychology in Tel Aviv University and then her PhD also there, and conducted postdoctoral research at Stanford University with Brian Mandel. She studies brain systems that underlie language processing in healthy and atypical populations by combining cognitive measurements, diffusion MRI, and fMRI. Her research focuses on the development of language systems in children and in adults. In her research, she quantifies structural properties of these pathways with diffusion MRI and relates them to cognitive functions through behavioral and fMRI measurements in individual participants. We're very pleased to have you today, Michal. Welcome to our seminar.
Really?
Sharon, thank you for the introduction. Did you want to say anything else? I think I muted you by accident. Do you want to unmute, Sharon?
Was my introduction heard?
Most of it, pretty much all of it.
Oh, okay. I'm sorry for this. I can repeat it.
But... No, please don't.
Okay. I wasn't aware of that.
I'll take it on. Thanks for the invitation, and thank you everybody for joining. And let me share my screen with you. There we go. Okay, what to do this? Good. So you can see my screen okay?
Yeah.
All right, great. So the title of the talk is Surprising Generalizations in the Neural Implementation of Hebrew and English Word Reading. And I planned this talk a long time ago. I think I gave you the title probably last year when we were planning this talk, and it was supposed to be a real talk, not a Zoom talk. And then things changed on us. I see that this is not going to work. And then just this morning, I realized that without ever knowing, we scheduled this talk to be exactly on the Hebrew Language Day, which is the birthday of Eliezer ben Yehuda. So I think this is a wonderful co-occurrence or coincidence that we get to talk about Hebrew and English on this very day. And believe Sharon and I that we had no idea what we planned. But chance is working on our favor. So I wanted to start by looking at English and Hebrew side by side. And for most of you who are readers of Hebrew, if not all of you, I think it's a good practice to look at Hebrew reading and English reading in the wild, and try to zoom in on the different processes that we carry out when we look at these texts. And this is how we would like to study reading, focusing on multiple different processes that happen at the same time when we're trying to basically extract meaning and enjoy different aspects of the language. So while we're reading, there are plenty of things that we have to do. We have to maintain stable fixation. Otherwise, the pictures of the words are going to be extremely fuzzy and we're not going to be able to decipher them. We need to program efficient saccades between one word and the other. We need to recognize letters. We need to convert these letters to sounds. We need to recognize whole word forms. extract structural cues, access the meanings of the words, and then there are text level aspects that I didn't even mention. And my point in this slide is that there are many differences between English and Hebrew in how we carry out the specifics of these processes. And for example, when we talk about fixations, it's well known that when English readers are presented with words, they tend to fixate slightly left to the center of the word, if it's an English word. When we read Hebrew, however, we tend to fixate slightly to the right of the center of the word. So that's one clear visual differences. Of course, we move our eyes from left to right when we read English and from right to left when we read Hebrew. Hebrew letters are known to be more similar to each other. I have to change one thing, sorry. Something about this laser pointer is not working out for me. So some would argue that Hebrew letters are more similar to each other, and that poses an advanced challenge on letter recognition. We all know the curiosities of English when you try to translate between a letter and a sound, or a grapheme and a phoneme. There are many peculiarities in how you do that in English, and many irregularities in these conversions. In Hebrew, on the other hand, something else is happening, and we tend to write our words without spelling out the vowels in most cases. So that encompasses a whole other challenge, but it's mostly a phonological challenge. And today I'm going to focus on a different aspect of processing words in both of these languages. And this is the structural challenge. How do we decompose a word into its structural elements, morphemes? And we do this both in English and in Hebrew, but we do it slightly differently. As you can see here, and we'll talk about this much more later on, English is a linear morphology, and the morphemes combined linearly, whereas Hebrew is an interleaved morphology. So we take the root and we interleave it within a phonological pattern. And this is going to be pretty much the center of what we talk about today. So given all of these differences, can we still identify a neural generalization in the infrastructure of written word structure in Hebrew and in English? And if we do, I think it's going to be surprising. And my title has already disclosed that we are going to look at some generalizations, and I think they're surprising due to all of these differences. So we have to get into some models of language and brain in order to make this discussion more well-informed. And I want to start, since this is vision sciences, I almost said vision lunch, since this is a vision science forum, I want to start from the very beginning, because I really don't know what you know about language and the brain. So the very beginning are these very simple linear models of language processing and written word processing in the brain, proposed by neurologists in the late 19th century, Broca and Wernicke and Lichtheim and Desjardins. And all of these great neurologists constructed a very simple model of language processing in the brain and of reading in the brain. And the model is linear in the sense that the information only flows from, let's say, the primary auditory cortex here to Wernicke's area for comprehension, through the arcuate fasciculus to Broca's area for planning production, to the motor cortex where we produce speech. And so this is a very linear, very simple model. Desjardins basically added another component to it. If we want to read a word, then the whole information begins in the visual cortex, transferred to the angular gyrus. And then once the angular has transformed the information into language representations, we send it off to Wernicke's, and we have already solved that part of the puzzle. So we don't need to solve it again. So very simple and very linear. Things had to evolve at some point, and we have vision science to thank for the evolution of these models, I believe. So inspired by these models of the dorsal and ventral pathways for visual processing, and later on for auditory processing as well, people developed more complicated models, less linear and more divergent models of language processing in the brain for language and for reading as well. For example, in this model by Rauschecker and Sophie Scott, they propose a model of language processing in which you can see already both dorsal pathways and ventral pathways, where the dorsal pathways process some aspects of language and the ventral pathways process other aspects of language. I prefer to work with this model by Hickok and Popper, and according to this model, quite similarly to the one by Joe Rauschecker and Scott, the model suggests that there is a division between dorsal pathways that process the phonological information and map auditory speech into articulation codes, whereas the ventral pathways are more involved in lexical access, recognizing the words and accessing their meanings. So we have ventral going at the bottom of the brain for meaning, and dorsal going at the top of the brain for production and phonology, sound processing. Of course, in this model, you can already see that the other hemisphere is also involved. And keep in mind that this model specifically suggests that the ventral pathways are more bilateral, whereas the dorsal pathways are lateralized to the left. This is quite obvious in this model, and it will be an important piece of evidence for us later on. If we want to take this forward and think how this applies to reading specifically, because notice that this model is talking about speech processing, and we would like to discuss reading in at least most of this talk. So if we want to apply this to reading, this is a very useful model proposed by Stan DeHaan in his excellent book, Reading in the Brain. And DeHaan in this 2010 model already proposed that when we read, we involve the same dorsal and ventral pathways, dorsal access to pronunciation, ventral access to meaning, but similarly to Desjardins, DeHaan proposes that the information in reading begins from visual inputs in the visual cortex. And then, unlike Desjardins, DeHaan proposes that the converter, the place where we recognize visual words and send them off into the language system, lives here at the bottom of the brain, the ventral occipito-temporal sulcus. And this is the region that DeHaan calls the visual word form area. Other than this difference, the clear relation between these models is again the distinction between the dorsal pathways for phonology and articulation, and the ventral pathways for meaning and lexical recognition. And of course, we could make this model slightly more accurate if we remember that reading also involves the right hemisphere, also involves the corpus callosum for communication between the two hemispheres, also involves this cerebellum. So there's a lot more than this simple model, but it will suffice for now. So if we look at this, and I've been staring at this model for a while now, we've been asking ourselves, what about word structure? Where is in all this model the place where we decompose words into their different morphemes, into their structural units? And is it more related to the meaning aspects? Is it more related to the phonological, to the sound aspects? Where do we do morphology? And I want to explain a little bit about morphology, beginning from really starting off from the beginning. So when we talk about morphology in linguistics, and specifically in linguistics, we would talk about derivational morphology in this case. We talk about the internal structure of words. When we have complex words, like let's say unhelpful, we can decompose them into their building blocks that we call the morphemes. So unhelpful would be decomposed into unhelp and full, and each one of these components would add up to the meaning of this word, to the information that this word carries out. And morphology is a really powerful tool for us to create new words as well. We can create new words, we can understand new words like destamization or to unfriend, based on our being familiar with their morphemes. We know what friend is, we know what un means, and we can put them together and create new meaning in this way. Or continuing our earlier discussion about Twitter, to retweet involves a morpheme re and a morpheme tweet, and by putting them together, we know what this means. Morphology is a very useful tool in language. We have to ask, do humans actually decompose words as soon as they see them? And how can we quantify that? Is there a difference between different individuals in how well they do that, in how often they do it, and how fast they do it? And having done all of that, can we map this into specific pathways in our model of the reading pathways? That's what I want to do here. And in following the topic that we presented earlier on, we want to discuss it in different orthographies. So how does it happen in Hebrew, and how does it happen in English, and is there a generalization between them? I have to, again, make a little digression into how we measure pathways in the living human brain. And up until, I would say, 20 years ago, the main tool that we could use in order to do that was postmortem dissections. This makes our lives a little bit difficult for a few reasons. First of all, it's a little difficult to ask this brain to decompose a printed word in either English or Hebrew. And second of all, it's unclear in this brain, although you can still see the dorsal pathways, the ventral pathways, it's a little difficult to measure their quantitative properties. So dissections are mostly about visualization and less about quantification, at least in humans. And then since about 2000, we've been seeing more and more papers that use diffusion MRI. And diffusion MRI provides us with, today, the only tool to measure in healthy human beings, including children, the microstructure of the white matter in parallel to the same individuals carrying out cognitive tasks. And if they do that, then we can quantify the microstructural properties of the pathways, and then we can examine the associations between the microstructural properties of pathways on the x-axis and some cognitive measure of the individuals on the y-axis. And I'm going to swap these axes here and there. But in most of the scatter plots that I'm going to show you, there's going to be a pathway measure on one axis, a cognitive measure on the other, and we're going to try to see if we can relate between them. So basically, the way that we run these measurements, they're simple MRI measurements to run, and I'm just running through this very quickly because who knows who's listening. I have no idea how familiar you are with diffusion MRI. So we collect our MRI in the same machine that's used for functional MRI, typically 3T, but the sequence is different. And it's a sequence that's sensitized to the speed of water diffusion, just free diffusion of water molecules. as the subject is lying in the scanner doing nothing. They could be closing their eyes for all we care, or they could be watching a movie if it helps them stay still. They need to stay still through the whole scan, and it's about a 5-minute scan or sometimes 10-minute scan, depending on how many times you want to improve your images. So we collect these images and we weight the diffusion direction in multiple different directions, so that later on the trick is going to be quantifying the speed of water diffusion in multiple directions and then modeling which is the direction of the preferred diffusion and deducing from that what is the direction in which the fibers are going. So we're going to be able to extract the primary direction of diffusion, telling us where is the fiber going to. And we're also going to be able to extract microstructural properties depending on how much anisotropy, how much directional preference we can quantify towards the main diffusion direction. So basically, having quantified the directionality of diffusion, modeling it with a very simple tensor, extracting the eigenvalues of this tensor, we can calculate some measure of how similar these eigenvalues are. And this will tell us how much directional preference is there in any given point in the brain. So we cover the whole brain, and at any given part of white matter in particular, we ask, where is the preferred direction of diffusion? Is it going this way or that way or this way? And how much preference is there towards that direction compared to the other direction? And if we're deep in white matter, if we're in the mid-sagittal plane of the corpus callosum, there's going to be a very strong preference for diffusion to go left, right, right, left. But if we are somewhere in the ventricles, for example, there's going to be no preference, and the tensor is going to look almost spherical because diffusion is unbounded and it can go in many different directions. So anisotropy is basically a weighted standard deviation of these eigenvalues, and it quantifies how much directional preference is there at every given point. And this is going to be the main measure that we use as a measure that quantifies the microstructural properties of white matter compared to behavior. Alternatively, we can look at just the mean of the 3 eigenvalues, and that will also give us some information about the directional preference here. So don't forget that we want to look at associations with behavior, and so we have to measure cognitive tasks in the same individuals. And then once we've done that, if we quantified phonological awareness, for example, we can quantify some measures of microstructure and try to find associations between them. You can do this all over the brain. It's very common to do this voxel by voxel for the whole brain, or we can do this for a specific fiber tract, which we do very often. So the way we mostly use this this paradigm is that we use tractography algorithms in order to recreate white matter pathways in the brain. And then we quantify their properties and we look for associations with cognitive measures. The other thing that we do all the time is we look at the properties of these pathways, not at the average level, but actually along the pathway. And I'm going to show you some results of this type. So instead of looking for a correlation of the mean of the entire tract, we often look for correlations at every given point along the tract. And this gives us better resolution and sensitivity. We can run comparisons between groups in this way, and our lab has been doing a lot of group comparisons, for example, between adults who stutter and those who do not. Or we can look at correlations within the groups, and these could be groups of healthy, typical individuals, or they could be belonging to all kinds of clinical populations. So this is the basic tool that we're going to use. These days, we're moving along from using these very simplistic tensors to using more complicated shapes that probably describe more accurately the distribution of diffusion directions in these voxels. And this complicates our lives a lot, but it also provides us with better resolution, directional resolution. So that could be something that is advantageous. I'm probably not going to get very deeply into these methods at all. And most of the results I'm going to tell you about are coming from tensor estimates. Some are coming from more complicated measures like that one. Something to keep in mind, and then I'm going to open for questions if there are any questions about the method so far, that these parameters are going to be affected by many different structural properties of the white matter, including myelination, but not only myelination. So it's very likely that when we see associations between white matter measurements and cognition, they're going to be driven not only by myelin, but also by axonal density, perhaps also by the thickness of the axons, and very likely by the coherence of all the pathways within a voxel. So all of these have very strong effects on our measurements. So whenever we find an association, it could stem from some combination of all of these factors. It means that there's something in white matter that's different between these individuals, and it's associated with their cognitive abilities, but we cannot trace it down to one specific aspect of white matter most often. Right, so we're going back to talk about the project, but any questions so far about these methods that you'd like to put out?
Can I ask?
Yes, please.
Hi. Can I ask? This is what is the rate of processing? Is it like online analysis, or you analyze it after the reading? And
all the analysis that I'm describing here are done after the fact. So we collect all the data in the scanner. There are some scanners that can already do some analysis online, very basic, showing you streamlines online. Since the streamlines themselves, I do not really consider data, but just a useful way of localizing the white matter fibers. The online algorithms are of no real interest to us. So we take all the data that was collected later on to the lab, and then we run all the analysis that we're interested in.
And you rely on water diffusion? Sorry.
So the speed of water diffusion weighted by different directions is the data that we're analyzing here. So yes, it's the speed of water direction and of water diffusion, and it probes the structure of the white matter. But it's not enough to run diffusion weighted MRI. You have to run diffusion weighted MRI that is weighted along multiple different directions in order to extract these parameters. I hope that was clear.
Yeah, thanks.
Good. So we want to look at, we use these tools in order to study the pathways that carry out morphological processing. And in order to do that, we now need a proxy for morphemic processing. How do we quantify your ability to decompose a word into its morphemes? I'm going to explain this briefly. We use a paradigm that is called the morpheme interference effect. And this paradigm we did not make up. It's a very well-known paradigm that looks at different pseudo-words. All of the stimuli are pseudo-words, but some of these pseudo-words, so they're not real words, but they're made up, as you can see, from real morphemes, real components. And some of them are made up from one real morpheme and one invented morpheme. Others are made up from two real morphemes that just never combine to create a real word in language. So towerly is made up of two real morphemes, while towerla is created from one real and one invented morpheme. So it turns out that when you ask individuals to decide, is this a word or not? The judgment should be non-word in both cases, but it turns out that it's harder to judge this word as a non-word because it is made up of two real morphemes. And this is a well-known fact. And this is why the effect is called the morpheme interference effect. The fact that there are two real morphemes here interferes with your ability to judge this as a non-word. Okay, we're going to capitalize on that and basically measure the size of this effect for every individual. And this is going to quantify for us how sensitive you are to internal structure of words, pseudo words in this case. And as I said, this phenomenon is well known from as early as the 70s. And more recently, Davide Cripaldi has done a lot of work with this type of effect. And we actually got the stimuli for this experiment from him. And this was a joint project with Kathy Russell and Joe Taylor, who I will mention soon enough. So running this in English, the nice thing about this, so very quickly I said here that we can calculate the effect here either on the accuracy and just the difference in accuracy between these two classes of stimuli, or we can calculate it on the reaction time. It turns out that people are slower to recognize these creatures as non-words, and they also make many more mistakes on them. And if we just look at the mistakes here, you can see for 40-something individuals that they make many more mistakes on the real root stimuli, on the real morpheme stimuli, and they make many less mistakes on the invented morpheme stimuli. The main thing that you can see here is that there is a lot of individual variability. Some individuals are really affected by these manipulations and others not so much. This works great for us because, as you remember, we're looking for correlations. So we really need to have individual variability in order to find these associations. And so we're going to quantify this difference. If you look at the difference here, there is very nice variability between individuals in the, we call this the morphemic cost, the cost of the real morpheme in this type of stimulus. So the morphemic cost distributes very nicely. And now we're going to measure the properties of pathways in 45 individuals.
Michal, a question?
Yes, Rafi.
It seems that the ones that show the highest effect are the ones that are have poorer performance. Maybe you have a ceiling effect. I mean, let's say you expose it for a shorter time or something. Maybe you will get, can you comment on that?
This is definitely true for accuracy, not so much true for reaction time. And you can see the effect in both accuracy and reaction time. Well, I'm not sure, but having said that, maybe I'm taking it back that it's definitely the case. I'm not sure that it's all about the ceiling here, because there is still some variability in the maximum performance that they get to, even in the easy condition. It's probably not the case that it's all driven by the ceiling. Anyway, everything I'm going to show generalizes between accuracy and reaction time, so I think we're good.
Okay, thanks.
All right. So we have results from 45 English readers. The data was collected by Joe Taylor and Kathy Russell, as I said, in the Royal Holloway. So these are all English readers who are clean English readers, in the sense that most of them don't read other languages, and definitely they don't read Hebrew. So we're very lucky in this respect with the English readers. They collected this data as part of a much larger study, we asked them to add diffusion MRI measurements to the protocol, and this generated a really nice collaboration between us that is still developing. So they measured morphological sensitivity on all of these individuals with the morpheme interference experiment that I just showed you, and they measured their diffusion MRI, and we analyzed it. This was part of Maya Yablonsky's dissertation. So briefly, here are the results, and finally, a results slide. Took us a while to get here, but now we're well prepared to show many results. So I'm going to run through many different results, and mostly they tell the same story. The story is we find associations between morphemic costs, no matter how it's defined, reaction time or accuracy, and the properties of white matter pathways, specifically bilateral ventral tracts. And you can see in this particular case, one tract known as the IFOF, inferior fronto-occipital fasciculus, in the left hemisphere and in the right hemisphere. And along this pathway, we can find a large cluster, passes significance, that shows a significant correlation with morphemic cost. The correlation here is found between anisotropy of this pathway and the level of morphemic cost. So this general pattern of finding is something that repeated itself in this data. And we found these correlations in several different tracts, not only in, this is a really nice, clean picture of the IFOF, left side, right side, pretty much the same location of the cluster in both of them. Really nice to find such symmetric findings for the association with morphology. But the picture is not exactly as clean in the sense that we find these associations in several different pathways. The common thing to all of these pathways is they're all ventral, and they're typically bilateral. Okay, so ventral pathways bilaterally show associations with morphemic cost. And sometimes the measure is mean diffusivity, sometimes it's FA, sometimes the morphemic measure is reaction time, sometimes it's accuracy. It sort of generalizes, but it generally is the case that they are bilateral ventral tracts. In all these cases, as I said earlier, we look at both the ventral pathways and the dorsal pathways here in blue and green bilaterally. And we do not find the associations in the dorsal pathways. We do find them in the ventrals bilaterally. This should remind you of the Hickok and Popper model, in which the dorsal pathways were left lateralized, the ventral pathways were bilateral. And this fits very nicely with the idea that we find the associations with morphology ventrally bilaterally. It also suggests that there is some relation here to either lexical access or semantics or both, because that's what we think the ventral pathways are carrying out when we read words. So all of this happened in English. And at the same time that we were doing this, our approach, our goal was always to study this process in Hebrew. But in Hebrew, we had to develop everything from scratch, as is usually the case in Hebrew. And we developed a parallel way of measuring Hebrew sensitivity to morphological structure to the English one that we just described. Now, what's complicated about Hebrew, as we already alluded to before, is that there is the morphemes are interleaved inside the pattern. So instead of a linear combination of morphemes side by side, in Hebrew you have a triconsonantal root, and it fits into the pattern in an interleaved manner. Hebrew is also known as a morphologically rich language, which means that there is a very large fraction of the words in Hebrew that are morphologically derived or complex. For example, all of the verbs in Hebrew, all of them are derived from roots. This is not true for nouns, but it is true for verbs. Morphologically rich is just a way to describe this complexity. It's also a deep orthography because we don't spell out the vowels. So there are many things that are different between Hebrew and English. And those may affect the effects that we can describe in Hebrew, and they do indeed affect some of the effects. For example, a very well-known morphological effect known as transposed letter priming, orthographic effect known as transposed letter priming. In English, you can take a word and swap two letters, and if the letters are in the middle of the word, it's going to be a fairly effective prime for the word itself, even though you flip the letters. In Hebrew, this doesn't work, and it probably doesn't work because of morphology. So if you swap two root letters, there is not going to be a significant priming, even though this is supposed to be a parallel to English, but it doesn't pan out. What this means is that we cannot take for granted that if anything was found in English, it will generalize to Hebrew. And so we had to test. And we put together a test that tries to emulate the English morpheme interference effect, but now in Hebrew. So the way we do this is we take real roots like pei, tav, resh. We put it inside a real pattern, but it generates a non-word, maftera. On the other hand, we take invented roots like samech, daled, lamed. We put it inside a real pattern, and it ends up a non-word. So both of them are non-words, similarly to the English non-words. This is supposed to be the parallel. It's supposed to be more difficult to say that maftera is not a word compared to masdela, because maftera incorporates a real root, whereas masdela incorporates a non-root. Okay, and indeed, when you run this on a group of people, you can see that their response times are slower for maftera style stimuli compared to masdela stimuli, and they make many more mistakes. Their accuracy is lower for maftera compared to masdela. So this is a strong effect. It's not a tiny thing that you can sometimes see at the group level. You can see this in individuals, and you can see that the effect is large. We can drop down your performance to sometimes even 80% accuracy by using these real roots inside real patterns to create non-roots. In the actual paper, we also show that this generalizes between noun-like stimuli and verb-like stimuli for individuals. So this is a nice paradigm. Okay, so it's a nice paradigm. You have to make sure that you equate these stimuli for many different properties. So the effect is not about being more similar to a real word. It's about having a real root inside. And we did that. And then we measured this in 45 individuals and saw this beautiful variability similar to what we've seen in English. And again, in reaction time as well. And we measured the fiber tracts in all these individuals, every individual separately, okay, 45 individuals. We always do the fiber tracking individually. This is a completely independent data set in the sense that it was collected in a different scanner, in a different country, with a different sample of people who are all Hebrew readers. And as Hebrew readers, yes, they read English as well. Okay. So I'm going to tell you the very end of this story. The results were very similar, surprisingly similar. It took a while to get there, and the Hebrew results came through probably a little over a year than after the English results. But if you look at it closely, we find surprising similarities, in my view, between the Hebrew results and the English results. There are some differences in the details, but if you move a little bit back and look at them, you can see that there are correlations between morpheme interference and the properties of the pathways in bilateral ventral pathways, very similarly to what we found in English. Questions so far?
Yes, I have questions, but I can leave them for the discussion as well.
We can take a question.
Okay, so I want to ask, I see that the strength of the correlation varies across the fiber. And I understand that at some point there are joining fibers, and then there are fibers who are leaving this major tract, but I mean, there are areas where there are the same fibers. My question is how can you explain the variability between the correlation levels along this area, which looks to me, I don't know, but only from this angle, as like similar fibers in the same bundle. So why would there be such a difference in the correlation?
It's a very good question. Let's start with a really good question. I can only offer some guesses. Part of it is probably adjoining fibers, fibers leaving, fibers joining in, and changes in the morphology, in the overall geography or general shape of the tract, along the tract. And those might have a contribution to this. So it's possible that we need the bulk of the pathways in order to see the correlation, or we're measuring contribution of some of the pathways that adjoin and not all of them. That's one possibility. The other possibility that became clear to me in recent years is there are some studies of myelin. I don't think most of this is driven by myelin, but I'm going to throw it out anyway. Myelin levels do change along the pathways to some extent, even dynamically so, but they do. So myelin doesn't have to be smooth or equally distributed along the pathway. And I'm not even talking about the endings where everything diverges and so on. Even along the bulk of the pathway, it doesn't have to be distributed very equally. Having said that, I believe myself the first explanation a little more than the second. So I do think that this has to be with the shape of the tract and so on. What I want to highlight that I don't think this has to do with the cortical regions that are nearby to the tract. I don't think this has anything to do with that. And this data is sometimes interpreted in this way. If you find a frontal correlation, does this mean it's more about the frontal endpoints? I would highly doubt that. And definitely, if you find the association deep in white matter, as we do here, for example, I don't think it has anything to do with the properties of the insula that's nearby or anything like that. We are in white matter. If we move laterally to the insula, anisotropy is going to drop so low that we're not going to be able to measure anything of interest, or we shouldn't anyway.
Thanks, but you do think that this has to do with some regions that are reflecting these different, I mean, some activation, or there is, not only in the tracts, there's probably some manifestation of these differences.
I think we agree that the tracts are connecting regions in cortex, and they're connecting many regions. Okay, these are huge tracts. They're not connecting point to point. Big bundles of many axons that are connecting large territories in the occipital and temporal lobes with large territories, in this case, in the frontal lobe. Okay. So if the tract properties are different in some individuals, hopefully this has the effect that the communication between these territories is more efficient or less efficient in other individuals. beyond that, I wouldn't overinterpret it, because we're talking about very large tracts, and it's very difficult, I think, to deduce which points do they between which points they communicate.
But to follow on Sharon's question, is there any evidence from bold fMRI of a correlation of a similar sort somewhere in the brain, in the cortex, in language areas? In other words, correlation between this task and bold fMRI activation?
All I can say is we tried, and we couldn't find it.
Wow.
But it's not like we ran the exact same task in the scanner. We did something else in the scanner. Anyway, I'm just gonna mention that our efforts so far to find a correlation with the morpheme interference effect in bold activation were not successful.
Thanks.
May I ask something else?
Yes.
Okay, about Sharon and Rafi were asking about the tracks. I want to ask about the task and about the conceptualization of the morphological processes in them. So how do you conceptualize the ones who showed the larger morphological effects? I'll tell you why I'm asking, because we have a lot of tasks of lexical decision of pseudo-morphologically complex words or non-words that are morphologically complex. And you could see that some of them would say maftera, it's a place where many people solve a lot of problems. So actually the ones who are more creative maybe or have more poetic abilities might be the ones who show the larger morphological effect. So I was wondering how you conceptualize the properties of the ones who show the larger effect.
So excellent question. I'm going to use it as a leverage to move on also, because I can take your point to mean these are all really interesting results, but we want to make sure that we're not studying morpheme interference effects here, but we're actually studying morphology. And morpheme interference effects have these very unique properties, right? There's an interference effect here. So if you're better in inhibiting the irrelevant aspects in this case, you will perform better. So some of the individuals that are performing better, maybe it's not that they're not sensitive to morphology, they're just better at screening irrelevant information. Others are more sensitive to morphology, they're going to do worse. And then others maybe are more creative. I don't know, I don't even know where to begin. So we want to make sure that this is not just about morpheme interference, but actually about morphology. And to do that, I'm going to skip a few slides forward and then go back maybe to them. To do that, we also run this type of experiment with these individuals where the behavioral task is completely different. And in this behavioral task, and this was recently just published a few weeks ago, they listened to a word, in this case, finally a word and not a pseudo word, already a relief. So they're listening, let's say, to tizmoret, and then they have to produce as many words as they can from the same root. They have 30 seconds to do this root-based fluency task, because to tell you the truth, after 30 seconds, they run out. Actually, they run out pretty quickly. It's a relatively difficult task for them. Unlike semantic fluency, people dry off very quickly. And then you count how many, what's the fluency level in this task. And it has something to do with their fluency, but it also has something to do with their sensitivity to roots, because we can run the analysis controlling for other forms of fluency, like fluency based on a semantic cue or phonological cue. And to make a very long story short, we generalize the findings and we find bilateral associations in the ventral pathways for this task as well. This we also generalized with different processing streams completely. So we use these more complex shapes to model diffusivity. We use probabilistic tracking, which is the more commonly used method today. We run the analysis actually in both ways, the deterministic with tensors and the probabilistics with constrained spherical deconvolution. all of this is to say that there was a very nice generalization across tasks, across modalities, because this is all auditory and production. This is not about reading, or at least we didn't ask them to read. Maybe they imagined written words. And we see the same bilateral ventral correlations with this task as well, with root-based fluency. Having said that, there are also some fluency-related pathways that show an association now. And this is a task that I'm particularly fond of, the frontal Aslan tract, and it connects Broca's area with the SMA, and it has something to do with speech rate and fluency. And it showed a correlation with this task as well. So it's possible. So task has an effect, and I think this is an important thing to say as well. It's not all about the core processes. Task can modulate the type of associations that we measure, and hopefully this answers to some extent. I do want to go back to something I mentioned here in passing. We ran another study with Yoram Boneh and Uri Polat that extended the morpheme interference effect to another task and another dependent measure. And we did this by measuring microsaccades, or actually Yoram measured microsaccades, and the microsaccades were measured in response to pseudo words that had invented roots, pseudo words that had real roots, as well as words. But the subjects were not asked to actively decide for every stimulus if it's a word or not a word, but rather only if it's a word, they have to press a button. So if it's a non-word, they don't do anything. And the advantage of that is that you can measure the microsaccades without any motor response. And while doing that, it turns out that there's a significant and rather robust difference between the patterns of microsaccades, the time, the microsaccadic rate, and so on, between these two conditions. So this is a nice finding that sort of generalizes the behavioral effect that we've seen earlier. It was only done in Hebrew, and if anybody wants to run this in English, I think this would be really nice. We're currently trying to replicate it for auditorily presented words with the help of Oren, who I think is here in the audience, and Yael Gera, who is running the experiment, and Yoram as well. All I can tell you is that auditory words are much more complicated to work with compared to visually presented words, because for some reason they unfold over time rather than being presented all at once, and it really makes our lives much more complicated these days. But so I wanted to mention this very quickly, and I can only very briefly say that we do find some generalization between modalities in the behavioral aspects of morphological decomposition, so that the morpheme interference that you find for visually presented words also generalizes to the auditory domain, to auditorily presented pseudo words. But I'm not going to get into the details of that. All right, so I want to summarize and close by saying that what we've shown is that we find word structure associations in bilateral ventral pathways, both in Hebrew and in English word reading. And this is a nice generalization across different tasks, different modalities, different processing streams. And it seems to support the idea that the sensitivity to morphological structure, and more specifically root extraction, this is what we've been talking about all along, root extraction, might be an essential component of lexical access, which is currently what we think is implemented by the bilateral ventral pathways. I do want to close with a cautionary note, and that relates also to my answer to Sharon earlier on. we look at these correlations, and it's tempting to interpret them as if they indicate a one-to-one mapping between a tract and a function. And I think we've been doing this in fMRI long enough to realize that we shouldn't. This would be a mistake because we're talking about very large pathways, and they don't have just one function. They connect between large swaths of cortex and distinct parts of the brain, and they probably relay a lot of information, different types of information between these distant parts of the brain. So I think the fact that you find one association, even if it recurs over and over again, probably does not mean that this is the function of this tract, but rather that this is one function that this tract is facilitating or modulating. And there are many other tracts that the bilateral ventral, many other functions that the bilateral ventral tracts are involved in. So that was just a disclaimer that I wanted to include at the end. And I want to say thank you to many students in my lab and collaborators, but particularly to mention Maya Jablonski. Most of what I described to you was her PhD, and she's now at Stanford working with Jason Yateman for her postdoc. And thank you all for listening.
Thank you very much. I want to ask everybody to unmute yourself, and let's give a big applause for wonderful, very interesting talk. And I'm opening the stage for Hagit, you want to ask? Okay, so I'm opening the stage for questions. Anybody who wishes, to ask, you're welcome.
Okay, I have a question. I wonder if you compare across individuals the changes along the fiber track. So ignoring the functional aspect, just the anatomical aspect, do you see correlations? In other words, some parameters, let's say the variance or the strength of the effect or whatever, does it correlate across individuals anatomically? You see what I'm asking? If you plot sort of a profile of some parameter, let's say the parameter of the strength, but you compare
the profiles are very consistent across individuals. Of course, they vary in the overall values, but the shape of the profiles, which is something we look at individually, is generally consistent.
So my follow-up is, for those regions that show the correlation to the functional, was there some peak in some aspect, in some
I can tell you
maybe variance, maybe strength, I don't know. Because it seems that the effect is localized to a specific segment of the...
But not exactly the same segment all around. For example, if you want to get into these details, I'm not sure we do. Look at the correlation here in the IFOF. This is in the posterior part of it, sort of. This is with the spoken task. If we go to this task, well, it's a larger cluster. But I would say it's probably a little more anterior. So this is probably taking it a little too far. But all I can say is, at least with respect to this kind of effect, if you see the cluster at the depth of the pathway and pretty much where the pathway becomes narrow, then you know that the anisotropy is probably picking up somewhere around this area because all the pathways are more packed, and density makes anisotropy go up. I don't know, generally the shape is more consistent, so the directional coherence is also affecting anisotropy positively. So this is a part of the track that generally has higher anisotropy compared to the endpoints, let's say.
Okay.
Mostly I use this in order to say that if you find an effect that's really at the end points of the tract, you don't want to make a big story about that, because the anisotropy is not very reliable as you get closer and closer to the cortex. So in general, we don't even bother analyzing these tracts when they get closer to cortex. And you can see that our analysis is only focused on the central segments of the tract. And that's just us trying to avoid these areas where the measurements are not very reliable.
Okay, thanks.
In the past, so in some of these tracts, this is not a very major worry. In the IFOF, I don't think it's a very major thing to worry about because, well, it jumps a little bit, but it's not dramatic in the sense of anisotropy going up and down along the tract. In the arcuate fasciculus, this is a major concern because in the arcuate, and this, it's a whole language talk without talking about the arcuate. This is pretty much the first time I'm saying the word. I'm very proud of this. In the arcuate fasciculus, it diverges into two separate branches, one that goes to the temporal lobe and one that goes to the parietal lobe. And around that point of divergence, there's because there's a split, there is a dip in anisotropy, and that's a meshulash Bermuda that you want to avoid. You just don't want to find your findings in this Bermuda triangle of the arcuate, because at that point it might just reflect a different fraction of the fibers going parietally and temporally or something like that. So you want to make sure that you're in the actual fiber tract that you're trying to measure.
I'd like to follow up on what Rafi asked. Maybe you actually said it, I'm not sure. So it could be that the peak of the correlation is at the narrowest, or the area where everything is the most similar across individuals. So maybe the issue of why the correlation drops is because there's more variance between the fibers across individuals.
It's possible. Actually, if this is the case, then I think the methods are working. Okay, so
yeah, definitely.
If we find the association where anisotropy is high, where we know that we're measuring the properties of these fibers that we targeted, then that's where we want to be. The concern is where you find it in the less reliable parts of the tract, and then it could be a contribution from another pathway just going by or something like that. So in principle, when you find it deep inside the IFOF like here, then you're pretty safe, I think.
Yeah, no, definitely. I was just wondering if that could be one possible explanation. No, definitely.
It could, even though many times, if you have a source that makes all the subjects more similar in a certain area, this is not where you're most likely to find the association. But in terms of the reliability, you want to be in the part of the tract where FA is generally high, and you think that you're measuring the properties of this tract and not another tract going back.
Thank you.
If there are no other questions, I can very briefly show you or just give you a hint towards other types of similarities that we find between Hebrew and English. I just had to explain too many things in order to show this, and this is a completely different line of research. There's a whole literature that shows measurements of population receptive fields. When you do this in the early visual cortex, you'll find something like this. V1 would prefer stimuli in the contralateral visual field over stimuli in the ipsilateral visual field. You can take these measurements and you can run them in the visual word form area. And then you can do this in Hebrew and English. We've shown, this is already published in Journal of Vision, that if you present words in English, then the visual word form area would, broadly speaking, would prefer words that are presented foveally and slightly to the right of fixation. So it has some contralateral preference. Once you look at this result, the next question you ask is, what happens in Hebrew? Is this all about the direction of scan, direction of moving your eyes, the attention to the important part of the word? What is it about? Or is it about properties of these brain areas that are generally contralaterally preferential? And when we did the same measurements in Hebrew, we found really striking resemblance in the measurements in Hebrew and in English. And so the part of the brain that's relatively selective to written words in Hebrew as well, when it's in the left hemisphere, it prefers slightly contralaterally biased words. And when it's in the right hemisphere, I can tell you that it has the opposite shape. So it seems to inherit its contralateral preference from the earlier visual fields, or definitely not a mirror image of what we found in English. This is a whole other stream of studies that shows a really nice generalization between English and Hebrew, and seems to measure properties of the, inspired by properties of the brain rather than the type of script that we read. Having said that, all of the participants that we study in Hebrew are always have been exposed to some English in their lives. And you can say that about the results that I showed you earlier, as well as about these results. We were never able to measure participants who read Hebrew well and have never been exposed to English, or I don't know, do not have very good English exposure. And so it's possible that there is some kind of effect here. I prefer to think that this is because this region is in the left ventral occipital temporal cortex, and regions in left ventral occipital have some bias towards the contralateral visual field. All right, so I just thought I'd...
Yeah, that was wonderful.
All right. All right. So any other questions, or I can let you all go and have your lunches?
I thought somebody wanted to ask a question, but if not, then let's all thank again Michal for a wonderful talk.
Great
really
talk, Michal. Thanks.
Yeah, really great talk. Very clear. And yes, and we'll meet here again next week. We have another talk. I will distribute that, so you're welcome to join us. Thank you again.
Thanks, everybody. Bye-bye.